Hello. Now we are going to turn our attention to the anatomy and physiology, really the anatomy and reproductive endocrinology of reproductive systems, starting today with the male reproductive system. So we will talk about anatomy and pituitary hormones and steroid hormones that are important in the male reproductive system. Before I go on, let me remind you to look at Moodle. There is a post there about my plan for the schedule for the rest of the semester. So here are two images of the male human reproductive system. This image here on the right is a midline sagittal section through the male pelvis. So here is the pelvic bone. Right here is the pelvic bone. Here is the anus and the rectum. So this is posterior back here, and this is anterior here, just to get oriented. So let's talk about some of the anatomy of the male reproductive system. The testes, just to be clear, the word testis is singular, testes is plural. The testis, in this case, one testis, is located inside the scrotum. The testis is attached to another structure called the epididymis. The testis is going to produce sperm and semen, and the mixture is moved through the vas deferens. The vas deferens is the tube that the sperm move through. The sperm, not semen, because the semen is produced in the prostate gland. So the non-sperm components of semen are produced in the prostate gland. The mixture of semen and sperm it's produced pretty much all the time, and that mixture is stored in seminal vesicles, in a seminal vesicle. There must be some way of controlling it, because if the seminal vesicle gets too full, it would be a problem. So I don't know what it is, but there must be some way of removing the semen and sperm mixture if it gets to be too much. In any case, the semen and sperm mixture that is stored in the seminal vesicle is stored there, but during ejaculation, it goes out through the urethra. The external structure, as you know, I'm sure, is called the penis. The penis has some internal structures some specialized names and internal structures that I do want you to know. The glans penis is the name of the tip of the penis. The prepuce is the Latin name for foreskin, which is sometimes removed in childhood or at another time during circumcision. The internal structures, the most important internal structures for the penis, are two vascular structures, one called the corpus cavernosum and the other being the corpus spongiosum. These are spongy vascular tissues inside the penis. This is the corpus cavernosum, and this here is the corpus spongiosum on either side of the urethra, which I'm having trouble drawing now. Those are highly vascularized tissues that are going to be important for the male erection. Ejaculation is caused by a spasm, by a contraction of the bulbospongiosus muscle. So that's what there's a muscle here. Women have it too. The bulbospongiosus muscle is going to be required for ejaculation. So this slide has all of the terms that I want you to know for the male reproductive anatomy. Not all of them, no. We're going to have another slide that will get into some more detail in the testis, but this has many of the terms. And I've highlighted most of them, but all of the terms on this slide, except for the ones related to urination, those you do not have to know. Okay, so the terms related to urination, you don't have to know, but the other terms you do have to know. This over here is an anterior view of the male reproductive system, showing really the penis is sort of transparent here, but it shows the organization of the testis. Let me get a different color. The testis, the vas deferens, which goes behind the bladder to the prostate gland. And so the prostate is kind of behind the urinary bladder. This tube, the vas deferens, goes from the testes into the abdomen by a structure called the spermatic cord. Here is a slide that shows internal and more detailed structure of the human testis. So the outside layer of the testis, the outside layer, is called the tunica albuginea. The tunica, the white tunic, tunica albuginea. The testes themselves, each testis is formed of a set of lobules. So this is a lobule. In between the lobules are septa, which is just Latin for wall, or septum. Inside the lobules are really the functional unit here for the production of sperm. Inside the lobules are an important structure called the seminiferous tubules. Each lobule has a set of seminiferous tubules. That's going to be where sperm is made, in the seminiferous tubules. The seminiferous tubules each has sort of an exit point here into a mesh, which is inside the testis, called the rete testis. The rete testis has a set of tubes that leaves the testis proper and goes into the epididymis, where they form efferent ductules. The efferent ductules merge into form the vas deferens, which leaves the epididymis to go into the abdomen to the prostate gland and then into the urethra. Sorry. So, functionally, sperm is going to be created in the seminiferous tubules. That sperm is going to go through into the rete testis. From the seminiferous tubules to the rete testis to the efferent ductules to the vas deferens. Here are two of the most important cell types in the seminiferous tubule. Well, in and around the seminiferous tubules. So here is a cartoon of a seminiferous tubule. So here is a cartoon of one seminiferous tubule. This one right here. This is one seminiferous tubule. Here is the lumen of the seminiferous tubule, and here is interstitial space outside of the seminiferous tubule. So the seminiferous tubule has cell layers, a lumen, and an interstitial space surrounding it. The cells in the seminiferous tubule itself, now zooming in, the most important cell type here is the Sertoli cell. And the spermatids, the stem cells that lead to the development of sperm. So these are mature sperm, and we will talk in a moment about the development of mature sperm cells from their beginning point as spermatogonium, then to spermatids, and then to the mature sperm cells, spermatocytes. So over here, the cell types that I do want you to know about are the Sertoli cells, which are going to be supporting cells for the production of spermatocytes. And we'll talk in the next slide about the details of spermatogonium, spermatids, and the sperm cells proper. Surrounding the seminiferous tubules in the interstitial space are a cell type called the Leydig cells. Your slide here shows them as the interstitial Leydig cells, but sometimes the interstitial is left off. They are always in the interstitium. The Leydig cells are outside of the seminiferous tubules. Another important concept that we're going to come to before the end of this lecture is the endocrine stimulation of the production of sperm. So the Sertoli cells are under control of a pituitary hormone called follicle stimulating hormone, FSH. Follicle stimulating hormone stimulates Sertoli cells to support the production of sperm. FSH is produced by the anterior pituitary gland in the gonadotrophs. They, in turn, are stimulated by GnRH, gonadotropin releasing hormone. So GnRH stimulates the anterior pituitary gonadotrophs to produce FSH and LH. The gonadotrophs produce both FSH and LH. LH stands for luteinizing hormone, and luteinizing hormone stimulates the Leydig cells to produce testosterone. So we're going to come back to that in a few minutes. This slide goes through the stages of the production of mature sperm cells in a process called spermatogenesis. So sperm cells start as spermatogonia. Spermatogonia are the primordial cells that develop into spermatocytes. Spermatogonia are stem cells, really. They are diploid. Now, I want to I want to talk about how this slide is laid out. So down here is the lumen of the seminiferous tubule, and out here is the interstitial space of the seminiferous tubule. And these columns, each of these columns is supposed to represent a Sertoli cell, maybe a set of Sertoli cells. Now, this slide shows the spermatogonia transitioning laterally as they develop. That does not happen. What happens is the spermatogonia transition in through the space of between the Sertoli cells. So this spermatogonium is going to develop and transition through here. And as it transitions into this space, it's going to become what is labeled over here, the primary spermatocyte. So this, what it looks like, when it looks like it's transitioning laterally, it's not. It's transitioning vertically, but they spread it out in space so that the cell types can be clear, because the cell types would be all stacked on top of each other if they did it in one of these spaces between the Sertoli cells. Bottom line is there is no lateral movement of the developing spermatids, but there is movement through the spaces through the between the Sertoli cells. Just going to clean this up a little bit. So this does not happen. So as a Sertoli cell develops, it develops first into, well, so it starts as spermatogonium, and then those spermatogonium develop into primary spermatocytes. That's shown here. Primary spermatocytes is shown in C and D. They are not over here. They are happening here for this spermatogonium. The primary spermatocytes, what's shown in C and D, the primary spermatocytes develop into secondary spermatocytes after they undergo meiosis 1. So meiosis 1, remember from CGI, meiosis 1 is the first step in the separation of the chromosomes in the production of germ cells, which is what we're doing here, producing sperm. So meiosis 1 generates the secondary spermatocytes from the primary spermatocytes. So these are secondary spermatocytes. The secondary spermatocytes undergo meiosis 2, becoming haploid, and those cells are called spermatids. Spermatids continue to develop the flagellum, the tails, the flagellum, and become mature spermatids or mature spermatozoa. So in the previous slide, I think I said that spermatocytes are the mature sperm, and that was wrong. Spermatocytes are a step in the development from spermatogonium to spermatids or spermatozoa. The names to know are spermatogonia as the precursor, primary and secondary spermatocytes, spermatids, and mature spermatids. And these mature spermatids are then produced into the lumen of the seminiferous tubule. Here are a set of slides that tries to illustrate what it is we're seeing in a somewhat less cartoon version. So here is a seminiferous tubule with the interstitial space surrounding it and the lumen in the middle. Here are the stacks, the columns of Sertoli cells. Here are the tails of the spermatids. And here you can see spermatogonia going through various stages of development as they develop into mature spermatids. So let's go over here. This slide is just slightly more elegant than the previous one. Spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and mature spermatids or spermatozoa, supported by the Sertoli cells. Outside of the seminiferous tubule, outside in the interstitial space, as shown right here, are the Leydig cells. Here is data. This is a histological section of a seminiferous tubule showing, let's see, here are the mature spermatids. I'm not sure I can tell the difference between all the different types of cells here. Here is a Leydig cell out here in the interstitium. Here are spermatogonia that are developing and moving. So there's spermatogonia that here have become spermatids and then are developing into, spermatocytes really, and then developing into spermatids here. Sertoli cells are shown in this slide as, but they sort of stretch long like that in order to support the movement and development of the spermatids from the precursor cell spermatogonia. Here is a scanning electron micrograph of a seminiferous tubule. So the whole thing is the seminiferous tubule. Here are the spermatids in the lumen. Even in the slide, they weren't sure which cell was which. So they're saying, okay, there's got to be some Sertoli cells here, but they're not sure which is which. So Sertoli cells and then the developing spermatids are in here, somewhere, going from spermatogonia to spermatocytes, primary and secondary spermatocytes, to spermatids. Going from the outside of this seminiferous tubule towards the lumen as the sperm cells develop. Now let's talk about the endocrine control of spermatogenesis. It starts in the hypothalamus. Hypothalamic neurons release GnRH into the median eminence, where it is picked up by the portal circulation and circulated to the anterior pituitary. The gonadotrophs of the anterior pituitary express the receptor for GnRH and express the enzymes necessary to synthesize LH, luteinizing hormone, and FSH, follicle stimulating hormone. Luteinizing hormone and follicle stimulating hormone are proteins. GnRH is a protein. So those are protein hormones. So here is GnRH production in the hypothalamus, portal circulation, GnRH in the portal circulation, GnRH going to the gonadotrophs of the anterior pituitary that synthesize follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone goes into the systemic circulation. So this is follicle-stimulating hormone in the systemic circulation, and this is luteinizing hormone in the systemic circulation. The cells in the testes express receptors for FSH and LH. Specifically, it's the Sertoli cells of the testes that express the receptor for FSH. And it's the Leydig cells of the testes that produce the receptor for LH. And one of the mnemonics I use for remembering this is that Leydig and luteinizing hormone both start with L. So Leydig cells are stimulated by luteinizing hormone, and then Sertoli cells are stimulated by follicle stimulating hormone. The Leydig cells stimulated by luteinizing hormone are stimulated to produce testosterone. Testosterone, as you know, is a steroid hormone produced by the Leydig cells in response to stimulus from luteinizing hormone. Testosterone is endocrine and paracrine. That is to say, it has endocrine function and paracrine function. Within the testes themselves, the testosterone diffuses locally to affect and to stimulate the Sertoli cells. So that is a paracrine stimulus. But testosterone also gets into the blood to cause some other changes, and that would be an endocrine function of testosterone. So together, let me just clean up a little bit here. So together, clean up a little bit more. So together, FSH and testosterone stimulate the Sertoli cells to stimulate spermatogenesis, the development of sperm cells from spermatogonia into mature spermatids, going through stages of primary and secondary spermatocytes. It stimulates the meiosis process and the cell division. So cell division of the spermatogonia and the meiosis of spermatogonia to produce mature spermatids. Both of those, all of that is stimulated by FSH and testosterone. Testosterone has negative feedback. So this is now endocrine function. It has negative feedback. Testosterone inhibits the gonadotrophs of the anterior pituitary to producing LH and FSLH, and it inhibits the hypothalamus from producing GnRH. So again, there's homeostasis, positive feed forward, positive feed forward effects, and negative feedback effects to try to maintain homeostasis of spermatogenesis. There is another hormone that I will not test you on called inhibin that is also produced by the Sertoli cells that does negative feedback. But the one that I want you to know about is the concept that testosterone, in addition to having positive feed-forward effects, a positive feed-forward effect that is paracrine, and positive feed-forward effects in the rest of the body, also has negative feedback effects on its own production. Let's talk some more about the hormones. So luteinizing hormone. Luteinizing hormone is a protein. It is actually structurally very similar to FSH. So FSH, follicle stimulating hormone, and luteinizing hormone have the same alpha subunit, but they have a different beta subunit. They're proteins. The stimulus for production of luteinizing hormone is GnRH, a peptide hormone from the hypothalamus, released into the portal circulation. The cells that produce LH, luteinizing hormone, are the gonadotrophs of the anterior pituitary, and the target cells for luteinizing hormone are the Leydig cells, the interstitial cells of the testis. The receptor for luteinizing hormone is a GS-coupled GPCR, and its function on the Leydig cells is to stimulate them to produce testosterone and other androgens. Follicle stimulating hormone is also produced by the gonadotrophs of the anterior pituitary. It is also a glycoprotein. It has the same alpha subunit as LH, but a different beta subunit. Its stimulus is also GnRH from the hypothalamus. FSH, follicle stimulating hormone, is produced by the Sertoli cells that make up most of the seminiferous tubule and surrounding the lumen of the seminiferous tubule. The receptors for FSH are also a GS-linked GPCR, and the main effect of FSH, follicle stimulating hormone, is to stimulate the Sertoli cells to help them produce sperm, increasing in sperm production, increasing in cell division of the spermatogonia, and increase in meiosis of the spermatogonia to produce spermatocytes and then mature spermatids. So testosterone is a steroid hormone. It is produced from cholesterol by steps of enzymatic production. So the precursors to testosterone, such as DHEA and androstenedione, are androgens. So these are molecules that are also hormones, but the most effective androgen is testosterone. All three of these, DHEA, androstenedione, and testosterone, are androgens, and they are all produced from cholesterol by sets of synthetic pathways. Again, I will not ask you to know the details of the synthetic pathway. I will not ask you to know the name of the enzymes that make them. I will not ask you to know the details of the pathways. I won't ask you to know the details of the pathways, but I do expect you to know that they are derived from cholesterol, that all three of these are androgens, and that they share pathways with other steroid hormones, cortisol, aldosterone, and the estrogens that we'll get to in the next PowerPoint lecture. So now let's talk some more about the steroid hormone testosterone. Testosterone is a steroid. It's produced in the Leydig cells of the testes. I've said that before. Which are stimulated. The Leydig cells are stimulated by LH, luteinizing hormone, from the gonadotrophs of the anterior pituitary. Testosterone has paracrine effects in the testis itself to stimulate the Sertoli cells, to stimulate spermatogenesis, but they also have many endocrine effects on muscle, on bone and in the brain. The receptor for testosterone are cytoplasmic. The receptors are cytoplasmic. They become transcription factors in all of the cells that express that receptor. So to be clear, the cells that express testosterone receptors are Sertoli cells and many other cells in the body, in the male body primarily. Well, that's not true. There are androgen receptors in the female body as well. Their effects on the Sertoli cell is to increase spermatogenesis, and their effects in the rest of the body on muscle, bone, and brain are to produce the male physical characteristics, such as facial hair, muscle mass, and affecting the brain of the male, affecting male behavior, risk-taking, and other forms of stereotyped male behavior. Of course, I'm stereotyping, but that is the stereotype of male behavior under the partial control of testosterone. So let's talk about male development for a moment. This is the structure of testosterone. So I will not ask you to recognize testosterone and differentiate it from any of the other steroid hormones. But again, you should be able to recognize this four-ring structure as being a steroid hormone. Came from cholesterol, and that's where it got this four-ring structure. In a male with a Y chromosome, there are genes that produce proteins called the SRY gene and the SRY protein. And these proteins trigger the development of the testes. They inhibit so-called Mullerian structures that are present in the female. So the SRY protein helps the Sertoli cells develop into testes instead of developing into the female Mullerian ducts. The Sertoli cells and the Leydig cells then, producing testosterone and other androgens, help develop the male external genitalia and Wolffian ducts as part of the male reproductive system. So what do I want you to know from this slide? I do want you to know that males have a Y chromosome. They produce proteins from a gene called the SRY protein. And those proteins lead to the development of Sertoli cells and Leydig cells that produce testosterone and lead to the development of male structures. You do not have to know the names of the Mullerian duct. So this name you don't have to know, this name you don't have to know, but there's some interesting structural changes that happen in the development of the male. So male reproductive biology requires an erection. So let's talk about the endocrinology and really, the endocrinology of the male erection. So it starts with arousal. You know what I'm talking about. This happens in the brain. So sexual arousal in the brain triggers the parasympathetic nervous system that you've learned about in this course to become activated, and the main neurotransmitter of the parasympathetic nervous system is acetylcholine. Acetylcholine released from nerves of the parasympathetic nervous system stimulates the blood vessels in the corpus cavernosum and the corpus spongiosum of the penis to release a local factor called nitric oxide. So acetylcholine from the nervous system stimulates the endothelial cells of the blood vessels in the corpus cavernosum and corpus spongiosum to produce and release nitric oxide. Nitric oxide then interacts with enzymes in the smooth muscle, the vascular smooth muscle of the blood vessels of the corpus cavernosum and corpus spongiosum, and it turns on an enzyme called guanylyl cyclase. You've seen that enzyme before in the retina. Guanylyl cyclase makes cyclic GMP. In the blood vessels of the corpus cavernosum and corpus spongiosum, cyclic GMP causes relaxation of the smooth muscle, vasodilation of the vascularization of the smooth muscle, and that allows blood flow to go to and stay in the corpus cavernosum and corpus spongiosum of the penis. And that's what triggers the rigidity of the male erection. The corpus cavernosum and corpus spongiosum become rigid with increased blood entrapment. At the end of the erection, cyclic GMP is broken down by a phosphodiesterase, a specific phosphodiesterase, phosphodiesterase 5. And so when cyclic GMP is broken down, there is less cyclic GMP, so there is less vasodilation, and so there is less engorgement, and so the erection subsides. Just as a point of interest, this enzyme is the target of the drugs that are used to treat erectile dysfunctions such as Viagra. So Viagra inhibits this enzyme, and so by inhibiting that enzyme, cyclic GMP is not broken down as much, and there is more engorgement and a longer lasting erection. I will not test you on the mechanism of Viagra, but it's interesting to know. Well, the erection is part of the story for male reproductive biology, but what we really need for reproduction is for male ejaculation. And not just anywhere, of course, it has to be male ejaculation into a female vagina. The mechanism for male ejaculation is complicated, but the things I want you to know are that it is the sympathetic nervous system and its neurotransmitter norepinephrine that is important. So the parasympathetic nervous system is important for erection, and the sympathetic nervous system is important for ejaculation. There is a nerve of the sympathetic nervous system called the pudendal nerve that innervates the bulbospongiosus muscle. And when sufficient stimulation has occurred to trigger the sympathetic nervous system to cause ejaculation, the bulbospongiosus muscle undergoes a series of contractions to squeeze semen and sperm from the prostate and from the seminal vesicles out the urethra of the erect penis. That's ejaculation. So let's finish with a couple of clickers. Just to sort of review and, I don't know, support your understanding. Which of the following are part of the pathway that leads to secretion of testosterone? So which of these leads to the secretion of testosterone? I'm going to pause and let you come up with an answer and then move on. GnRH from the hypothalamus and luteinizing hormone from the anterior pituitary are the correct answers for leading to the production of testosterone by the Leydig cells of the testes. Which of the following are part of the pathway that leads to the development of spermatocytes, or spermatids, really? I'll stop here, let you come up with an answer. All of them. So GnRH is important for the production of FSH and LH. The anterior pituitary's production of FSH is part of the story that stimulates the Sertoli cells to support spermatogenesis. LH stimulating the Leydig cells to produce testosterone, which in a paracrine fashion stimulates the Sertoli cells to support spermatogenesis, and the Sertoli cells themselves. So this was an all of the above answer. And finally, let me just tell an interesting story. You can put your pens down because I won't be testing you on this. This is a really weird story. So here's the abstract to a paper that was published in 1979. I'll just read it. In two separate procedures, a psychiatric patient first performed a bilateral orchiectomy on himself and then later attempted to denervate his adrenal glands. This case suggests that physicians should be alerted to the possibility of self-surgery occurring in patients that have sought elective surgery and have been rejected from elective surgery. So let me flesh this out a little bit more. This patient was a medical student. This patient was having a nervous breakdown and was aware that he was suffering from the early stages of schizophrenia. So he knew enough to know that he was not behaving in a typical fashion. He was not feeling typical. He was hearing voices. He was seeing things. He knew he was a psychiatric patient, but he was also a medical student. And so he sought elective surgery as part of his, because, well, what was he afraid of? So first of all, what is bilateral orchiectomy? Bilateral orchiectomy is castration. He asked to be castrated. And he asked to have his adrenal glands denervated. So, okay, so why is that? Why did he want that? Well, he knew that testosterone, being an androgen, causing male behavior, could lead him to become violent, aggressive. And he was afraid. He was not yet violent and aggressive, but he knew he was having a psychiatric break. And so he was afraid of hurting somebody. And so he said, well, if I want to prevent myself from hurting somebody, I'm going to castrate myself. So this is not very typical thinking, but you can sort of see the logic. Instead of seeking treatment for his schizophrenia, he was seeking treatment for the symptom, one of the symptoms of schizophrenia, which could be androgen-induced aggression. Well, so he, being a medical student, he had access to surgical tools, and so he performed a bilateral orchiectomy. He castrated himself. Now the question is why? So that's truly weird, but that was successful. The part that wasn't successful was his attempt to denervate his adrenal gland. Why would he want to do that? Well, he knew that the adrenal cortex is the source of cortisol and aldosterone. But he also knew that a small amount of testosterone is produced in the adrenal cortex because there is a small amount of the enzymes necessary to produce the androgens there. So he said, well, I can't just get rid of my testicles. I have to stop my adrenal glands. And so he sought to denervate, to cut the nerves that innervated his adrenal gland. That's difficult because the adrenal glands are on top of the kidney. The kidney is way up underneath the ribs behind on your back. So he had a hard time reaching it. And he set up a mirror system to see what he was doing, but he made incisions in his back and he accessed his adrenal. But in the process of cutting the nerve, he cut a blood vessel and he knew he was bleeding to death and he sought help. And that's when he went to the hospital for both fixing his surgery and for treating him for his psychiatric problems. So this is an interesting case that I use just to illustrate some of the concepts of male endocrine physiology and anatomy. That's the end of this class. Next time, we will talk about female reproductive anatomy, physiology, and endocrinology.